My research focused on two crucial ocular reflexes, the vestibulo-ocular reflex and the cervical-ocular reflex, which I'll refer to as the VOR and the COR for the remainder of this presentation. The VOR stabilizes gaze during head rotations by moving the eyes at the same speed but in the opposite direction of the head. For example of this, when you make an eye contact with someone while shaking your head no, you're using your VOR reflex, and the strength of this reflex is measured using something called the VOR gain, which can be calculated by dividing the peak eye velocity by the peak head velocity. The COR, on the other hand, stabilizes images on the retina during trunk rotations when the head is fixed in place. COR gain is measured by taking the ratio of the peak eye velocity to the peak trunk rotation velocity. And these reflexes, coordinated by the cerebellum, require inputs from the visual, proprioceptive, and vestibular systems to produce accurate eye movements. Now, individuals with subclinical neck pain, which I'll refer to as SCNP, experience mild to moderate recurring neck pain without treatment. Research shows that SCNP individuals have altered sensory motor integration and larger COR gains, indicating impaired cerebellar processing. The neck is a very important sensory organ since it contains a very high density of muscle spindles, which are basically organs that sense muscle fiber length and are therefore heavily relied upon for proprioception. We hypothesized that neck pain alters proprioceptive information received by the central nervous system, causing impaired sensory motor integration. And other studies have shown that neck muscle vibration can stimulate these same muscle spindles, and that can alter self-motion perception of vestibular origin. Now, this study aims to investigate the effect of neck muscle vibration on the baseline VOR and COR gains of healthy individuals and compare it to those individuals with subclinical neck pain. To test this, participants were fitted with an iLink 2 eye tracker and an active infrared marker to collect eye and head kinematic data. They followed protocols requiring them to maintain gaze on a visual target projected on a screen in front of them while performing head rotations in the VOR paradigm or being rotated in a chair with their head fixed in the COR paradigm. After baseline measurements, they wear blackout goggles and vibrating apparatuses are attached to their neck muscles. They sit in darkness for 10 minutes, followed by a second gain measurement. Then the apparatus vibrates continuously for 10 minutes at 80 Hz, followed by a third measurement, and participants perform the COR and VOR protocols at least 1 week apart. We're hopeful that this study will illuminate the impact of neck muscle vibration on the COR and VOR reflexes and provide valuable insight into the mechanism by which neck pain alters sensory motor integration.